Now that we're done with the centrifugation of our blood sample and we have the pellet and the serum, we may now prepare separately our samples for the proteins, the blood serum, and the cell lysate. So first, we need to aspirate carefully all of the serum from our centrifuge sample. So making sure not to disturb the pellet at the bottom. So we will aspirate this and then we are going to transfer this into another microcentrifuge tube. So this is our serum now. And then we're gonna continue aspirating the serum until we have aspirated all of the serum and only the pellet is left in our original tube. And then we'll dispense more of the serum into the separate tube. And here we continue removing the serum. And here is now our complete serum for SDS page later on. And now for our cell lysate sample, we need potassium chloride. And then we are going to dispense this with the pellet that was left a while ago when we removed all of the serum. Okay, this potassium chloride has a specific molarity, which when mixed with the cells, will cause the cells to lyse. And in order to make sure that the cells will lyse, we will be vortexing this mixture. And then we'll centrifuge our mixture of potassium chloride and pellet. This time at the maximum of 13,000 RPM for 5 minutes. Okay, so let's see what happened to our sample. So we can see here that the supernatant is actually not clear now. It's reddish, and we are expecting that the blood cells, usually the red blood cells, have lysed, and that the proteins are now here in the supernatant. So we will be aspirating the supernatant, leaving the pellet behind. And we'll transfer this to another microcentrifuge tube, and this will be our cell lysate sample. So take note, we are labeling this with RBC, but this is our cell lysate sample. So let's just make sure that we have aspirated all of the supernatant and transfer these to our cell lysate sample. So we now have two samples to be analyzed, the blood serum and the cell lysate sample.